Hi, everyone. Welcome to our Food Safety Office Hours. I'm your moderator, Sam Alkin, Associate Professor here in the Department of Food Science. The word of the day is Cyclospora. It's not just a great name for a summertime bike festival through strawberry fields, but it's also what you might bring home with you after eating strawberries from the fields where a bike festival passed through. So we spent a lot of time talking about foodborne bacteria and viruses in these office hours, so it's time we spread a little bit of the microbial love to parasites. And to help us kind of burrow into this topic today, we've got Dr. Kalmia Kniel from the Department of Animal and Food Sciences at the University of Delaware. She's going to start us off with an overview of the basic characteristics of Cyclospora and why we're still seeing outbreaks every summer. She'll also give some insight into what we do and don't know about possible reservoirs, routes of transmission, and how we as an industry might better detect and control this eukaryotic cousin. Then we'll have Amy Robbins, who's joining us from the New York State Department of Health's Bureau of Communicable Disease Control. She's going to bring us up to speed on New York State's parasite surveillance programs, recent updates to the program, and how state programs work together in a national enhanced surveillance network. And then lastly, we'll have Dr. Amy Kahler join us. She's an environmental microbiologist in the waterborne disease prevention branch at CDC. Give us a little bit of information on their view on Cyclospora, and then we'll open up the panel to questions from you from the audience. So as you're listening and you're thinking about those questions, as always, there's a little chat box down there at the bottom of your screen. You click that chat box, it opens up a little menu. You can type in your question, send it either directly to me or to the group as a whole, and then we'll answer those once we get to the panel. So keep thinking of those questions, and with that, I'll pass it on to Callie.
Okay, great. Thank you, Sam. That was a wonderful introduction. And it's such a pleasure to be here with everyone. And I'm very excited that, as you said, that we're going to be talking a little bit about the protozoa today in terms of food safety. So does that all look okay?
That looks good.
Great. All right. Oops, no. Okay. So as Sam alluded to, Cyclospora is not a bacterium. It's a protozoan parasite. So unlike the bacteria, protozoa do not grow outside of the host. They are transmitted, in this case, by a mature oocyst. And so we'll talk about what that means specifically as we go through the next few slides. While there are numerous questions that are remaining when it comes to control and propagation and infection of this specific pathogen, we're starting to know more and more, and I think we're at a really exciting junction in time, and you'll hear from the next two presenters as well about some of the interesting insights that we're really gaining. One point, though, remains is that we have some difficulties in studying this specific organism, Cyclospora, because of the lack of an animal and a cell culture model. But the good news is that we do have some surrogates that are quite promising. And so I'll share that with you. So that you can put this in perspective, I think it's important that we think about size, because when we talk about some of the potential mitigation efforts like filtration, size can really be important. So this is a eukaryotic cell, so I think it's more like our cells than like those of E. coli or Salmonella. And when we talk about Cyclospora, it's about 8 to 10 microns, so on the smallish size of a eukaryotic cell, but still much, much bigger than our viruses, of course, and bigger than bacteria as well, but still microscopic in nature. There are several protozoa that you're probably familiar with. You can see them kind of relatively depicted here in these cartoon images that were created by one of my grad students, now Dr. Shani Craighead. What we know about all of these is that they have a relatively low infectious dose and that they are resistant to our normal levels of chemical disinfectants. We'll talk more about that as we go forward as well. So there is a lot of interest in Cyclospora, and so it's wonderful that we're having this opportunity to answer questions and to talk more about it. The FDA within the past few years has published this Cyclospora Prevention, Response, and Research Action Plan that outlines what they identify as being necessary to prevention, also enhance response activities, fill in some of the gaps, CDC is also a big part of this, and they also have been able to look at a lot of research that has been funded by the Center for Produce Safety. And so it's wonderful to see many organizations getting on board here. Also, there was a charge that was presented to the National Advisory Committee for the Microbiological Criteria for Food, that's NACMIF, on how to prevent cyclospora contamination in food. And they put out a great report if you want more information on that. But all of this has really brought about more interest, and I think it's going to result in more funding, which always will result in more answers and better answers for us and for the food industry. So when we look at cyclosporiasis, which is the disease caused by Cyclospora cayetanensis, that infects the cells of the human intestine, and we'll talk about that next. We're looking at symptoms that can last 5 to 10 days or longer. In some cases, we do see and have record of individuals who have been sick for many weeks at a time. This most commonly is a watery diarrhea. Sometimes I've read notes of where it was explosive, but then you have all the other gastrointestinal issues, right? Loss of appetite, weight loss, cramping, bloating, gas, nausea, fatigue. This can be treated with some of the sulfa drugs, and you see the trade names there. So unfortunately, if you are allergic to sulfa drugs, that seems to be, the biggest conundrum there. Also, as we'll talk about, at least one of the outbreaks that we'll look at, when the FDA was able to identify that workers were being treated with these drugs, it was an indication that perhaps there could have been previous infections. And so that's something too that we can think about. Also, it is important to note that as with other foodborne illnesses, some people may be asymptomatic but still be shedding. So of course, we're always going to focus on good worker health and hygiene. So infected individuals will shed the unsporulated or immature oocysts, and that's here in the top left corner of this picture where you see this undifferentiated mass. So these are the top two are pictures of the oocysts. That's also depicted there in that cartoon image. And hopefully what you can see there is this really thick ring structure on the outside. That's a really thick, dense membrane that has multiple layers of proteins and carbohydrates there. And then also within the inside, there are two sporocysts. Those contain these sporozoites. That's that banana-shaped creature there on the right-hand side. The interesting and sort of the conflicting and confusing part of cyclosporiasis is that these immature oocysts are what are shed into the environment. Then those take some time, about 1 to 2 weeks, in the environment, and we still don't quite understand all the triggers that are necessary to make that immature oocyst become what we call sporulated or more mature, where you can see those differentiated sporocysts now on the inside. And when that happens, those can be ingested and they are infectious. They will, that oocyst membrane will rupture, will release those sporocysts, and then those sporozoites infect the intestinal cells. And that's what triggers your illness. Also, what's quite interesting about this organism and not completely well understood because of our lack of models. So we're really looking at a lot of its relatives to be able to understand this. When you ingest the sporulated, that mature oocyst, it infects the intestine. And here you can see on this nice image from the CDC, we have multiple asexual reproduction cycles that occur within the human intestinal cells and followed by sexual reproduction. And then the oocysts are shed in human feces. And so if you were thinking about, where are our aspects in terms of food safety, right? We go back to worker health and hygiene and also trying to eliminate having those oocysts appear on our products in the first place. So there are many challenges when it comes to thinking about what we know and where we can go with this information and identifying specific data gaps and challenges that are important to the food industry. We do know that there is a low infectious dose likely for this organism, similar to some of its related organisms, related protozoa like Cryptosporidium. It is very robust in nature and has this resistance to washing and chlorine, very much like Cryptosporidium and also like Toxoplasma. You've probably heard of those. I don't think there's necessarily a lack of awareness anymore. It's great to continue to talk about it and be thinking about it at the same time that we're recognizing risks with salmonella and Shiga-toxigenic E. coli, et cetera. But we're really trying to identify more aspects about the life cycle and also identify aspects that are related to detection and testing. And so that's where we'll go next. So when it comes to testing and detection and thinking about what types of tests can be done, at this time, I think primarily we're really looking at research purposes and also for testing and detection in outbreak situations and perhaps on recalled food products. I don't think we're quite where everyone can go out and do testing on their own and where it's yet recommended for industry to be doing that, although we're getting closer and there are some great tests that are being developed in collaboration between researchers and industry. But right now, some of the information, some of those results can still be a little bit confounding. So I think that's where we sit. We can have that discussion as we continue. So this methodology really is involving. We're looking at a lot at validation of methods on various food commodities, also looking at specifics that might relate to detection from soil and water, and again, looking for those oocysts and whether or not we'll be able to determine between sporulated and unsporulated oocysts is still another thing too that some scientists are working on. I love this quote about the evolution of testing that comes from my colleagues in the FDA and USDA, where there's great progress being made in the field of Cyclospora and the areas of molecular characterization of strains and species, generation of genomes, and development of novel detection methods. And that's really where we sit. And it's a very exciting time. There's a lot of information in particular coming out of the CDC related to the genetics of specific organisms, small genetic differences that might be identified in different outbreaks to help us better understand perhaps the root of those outbreaks. And so that's coming in the future. I think there's a lot of exciting science there. Again, we're at the tip of this all kind of coming to fruition. So let's talk more a little specifically about detection. The way I see it, we have detection in clinical situations, mostly done in hospitals or labs, right, when humans are sick. But then we have detection from produce items. And we have very specific validated methods through FDA BAM that says, which tests we should use and how those have been validated for specific produce items. Then we have the environmental detection, which remains still a little bit more of a complex issue, although we're also seeing more validated results come out for that as well. And historically, we've really looked at molecular detection using 18S RNA, which is kind of the equivalent for 16S in bacteria, if you're familiar with that. But because the Cyclospora has many related cousins, if you want to call them that could be in the environment, using just 18S alone was confusing. And I think Amy's going to tell us more about that in a little bit. And that led to perhaps some presumptive positives that maybe could not be confirmed. So what scientists are doing now is adding another target, this mitochondrial target that's known as MIT1C. And by using both of these targets in our detection systems, we're able to better detect and be more specific in terms of whether or not we have Cyclospora chitinensis there or not. And I think that that's where you're going to see a lot of new information coming out, and it's going to be very practical and very useful as we go forward. I'll also note that there are multiple mitochondrial targets that are being investigated by a lot of the clinical detection systems and looking at the epidemiology of some of the outbreaks. And in that area is also just growing in terms of the wealth of information. I think it's all going to be very helpful. So it still takes quite a bit of time for an outbreak to be detected, right? We're still looking at having enough samples and making sure that the proper tests have been done, detection has been done. And a lot of that when it comes to human illness relies on detection within labs. And the use is often of this BioFire gastrointestinal panel, which has been, I think, quite an incredible addition from my colleagues who are in clinical medicine. You can see it detects many, many different organisms that cause GI illness, including Cyclospora. And then if we want to go back to detection in produce or soil or water, that's where we're looking at the FDA BAM. And you can see there is 19B for detection on fresh produce and also 19C. And that's for water, excuse me. And these are continuing to be validated and expanded, and lots of good information coming there. So you've probably seen this slide before, which shows us this history of Cyclospora detection and identification. I want to show it again here to give you some context. When we think about Cyclospora, it's still relatively new to all of us. And so while many more scientists are working on it now, we're still trying to answer some of the basic questions. So let's jump through a few outbreaks to discuss, some of those findings and how we might use that to go forward. So if we look at the early outbreaks in the 1990s and early 2000s, we can see most of our outbreaks were associated in the US that occurred, outbreaks in the US and also North America occurred associated with imported produce. Things and crops were berries, snow peas, herbs, and lettuce. Also, some of the stealth ingredients like raspberry coulis or basil that might have been in the vinaigrette that you didn't realize was in the vinaigrette, right? All of these things may trace back very difficult. Also, we had less advanced molecular diagnostics, and now we have qPCR and digital PCR, which can really speed up and help with the detection. So now let's fast forward to 2013 up through the present. I like to show this outbreak because there was so much learning here. If you look at this epi curve here on the top right, you can see the 5-year weekly mean for cyclosporiasis cases was really low from 2008 to 2012. And then in 2013, we had this well-studied outbreaks in multiple states, either linked to salad mix or linked to cilantro. Those were imported produce items into the US, but you can see they had very clear epi curves there on the bottom. I like to show this slide as well because this paper is fabulous in terms of how it shows the complexity of the investigation for cyclosporiasis and the really detailed traceback analysis that was done in order to identify the food product. Of course, this was 10 years ago. So our traceback, while still being complex and difficult, right, we do have better traceability, and that's why that's so important for many growers to be able to know that their product was not involved in an outbreak, right? Just as much important as it is to know that it was involved. And so you can see here that many of these products were linked to restaurants as well, and so that also added to the complexity. This is the FDA assessment that I mentioned earlier, where they were able to go back and have really great open dialogue with the companies that were involved and shared information about the harvest crew in terms of what medications some of them were being treated with, and then thinking, well, perhaps they were infectious before, maybe not at the point when they were harvesting, but they had been treated with this. I show this only as a reminder of how important open communication within our workforce really is. So now we look from 2014 to 2017, you can see we have hundreds of cases each year of cyclosporiasis. These are happening during what's we call it a cyclospora season. I get that's not quite funny for many people involved, but it is a very seasonal illness where we're looking at illnesses that occur between May and September, or May and August. Here we're looking at laboratory confirmed case numbers. So we have many that were associated in 2014 and 2015 with consumption of cilantro. In 2014, there was a specific outbreak in Texas that at least they were able to identify many of the cases that were associated with that. But then in 2016, no reported connection or specific outbreak. And in 2017, again, no specific vehicle that was identified. And so that's kind of a trend. We're seeing more and more cases, but not being able to identify a specific vehicle. In 2018, we did have 2 specific outbreaks that occurred. One was linked to Del Monte fresh produce vegetable trays, and then also to salads that were served at McDonald's. The companies involved with these have been very forthcoming and done a lot of analysis in terms of wash water and other things like that to try to reduce these cases. Also, there is this term that's now being used by the CDC, domestically acquired cases. So these are individuals that have cyclosporiasis, it's lab confirmed, but have no history of international travel, again, during those months of May through August. In 2019, we have cases that were associated with basil. So we're still seeing these similar commodities, herbs, leafy greens. We haven't necessarily seen the berries recently that we saw early on. I think a lot of that has to do with keeping the cold chain, for example, and being very strict about hand hygiene and things like that. You can see we also have many cases here that go under that category of domestically acquired. We move forward into 2020 and 2021, the trend still continues. We did here in 2020 have an outbreak that was linked to bagged salads that had lettuce, carrots, and cabbage, and that were there was some investigation there by the FDA into water where the cabbage was grown in Florida. And then again, we have these lab confirmed cases that come under the heading of domestically acquired. 2022, again, we have sort of clusters that were identified by the CDC where they may have had consumption of leafy greens, but they weren't able to go back to a specific source. Again, it's kind of the same numbers that we've been seeing. And then 2023, again, these are during that Cyclospora season, May to August. And no deaths reported with any of these, although there are hospitalizations that occur each year. So where is this coming from and what can we do about it? So now we'll talk a little bit about that for the last couple of minutes. So here we're looking at the FDA herb sampling results that occurred between 2018 and 2021. And you can see a very small amount of detection there and Cyclospora in terms of herbs that were domestically acquired and also imported, right? And so the negative results there are the big white bar, the very small black line actually is the ones that were positive for Cyclospora. So given that, what do we do, right? We have lots of cases in humans and we don't necessarily have the detection on the product. But so what can we do to try to mitigate this? Since we do at this time still understand that cyclosporiasis is shed by humans, right? Cyclospora cayetanensis seems to only infect humans. There are scientists who are continuing to investigate if there's other reservoirs, but still we have this focus really on people. And so we think about our good agricultural practices, our education and training and open communication. But if we want to go somewhere else, where do we go for technologies for mitigation? And for Cyclospora, we're going to use surrogates. I've always been a proponent of using surrogates. I think they can be good models. I think we can learn a lot from them. So in this early work, you can see here that oocysts do attach. This is actually a toxoplasma oocyst, but about the same size and shape. They attach very well to berry surfaces, especially the little hair-like projections on a raspberry. Now we're using Eimeria as our surrogate. Eimeria is a very genetically related apicomplexan parasite too. It's very similar to Cyclospora in terms of its genetics. It's a little bit more of an oblong shape, as you can see here, versus the rounder Cyclospora. Eimeria in itself, in particular, if we look at Tenella and Acervulina, which you can see here on this phylogenetic affinity analysis, when we're looking at those two, they're actually really important when it comes to poultry production. Around the globe, they cost an average of $14 billion in terms of illness that they cause poultry. So by studying them, it's actually very useful to the poultry industry. And we know a lot of them because so many scientists have been studying them for a long time. So now let's go back to what we can do when we're using these surrogates. So one of the studies that continues to be investigated is the ability to filter. Can we filter out these oocysts, right? We think about their size. That should be a benefit to be able to have them removed. And so many growers are already using filtration systems. We've had scientists, including myself and members of the USDA ARS research community, funded by the Center for Produce Safety, to investigate whether or not adding zero valent iron to sand filtration can remove them. And we do see that in sand alone, we get a 2 to 3 log removal. If you add zero valent iron, you can increase that removal, but just looking at sand filtration alone, we get some removal, which is great. So what if you are not making just fresh produce, you're making some products? High pressure processing is one potential mitigation step. We have seen a greater than 5 log reduction at 85,000 PSI for you, which is the technology that's being used here in cold pressed juices that can inactivate Cryptosporidium. It's likely that it would also have similar effects on Eimeria. And if we look at what's happening with Eimeria, you can see about 79,000 PSI on treated raspberries and basil was enough to give a 5 log inactivation. So that's a potential for, berry purees and things like that. Also worked on basil. UV light had a little bit less inactivation in terms of raspberries and basil at 160 milliwatts per centimeter squared, or a little bit higher. So what's coming down the pipeline? I think that we're going to see some benefits from cold plasma and chlorine dioxide. We've worked with them in my lab on Cryptosporidium. We're now doing that work with Eimeria. We can use Eimeria in a human cell culture infection assay, which is very helpful. I know a lot of people want to know about water irrigation, irrigation water technologies. And here we're able to show that you need higher levels of chlorine and PAA to be able to inactivate Eimeria tenella. And so that's actually promising if we can get to those higher levels without causing more damage to the soil and things like that. So where do we go from here, right? I think we do have a lot of technologies that we can go to depending on the product. We can look at filtration, I think is very promising. I'm excited about a lot of the work that people are doing in sewage treatment. I can tell you that we've looked at wastewater analysis and we actually haven't found it even by using multiple targets. But I think you're going to hear a little bit about that more so in a few minutes. Key industry controls, though I still think need to rely on good sanitation, avoiding issues with water if there is a sewage leak, right? Sometimes those things happen and we don't know about them. We don't quite understand about the persistence of Cyclospora in the environment, so we're still really focusing on people. And then I think many of you probably already have these in your list of to-dos when it comes to health and hygiene with our workers, right? But making sure we have good open communication, proper training in the native language, when thinking about how to use toilet facilities, importance of proper hand washing, right? Because those oocysts can survive on toilet paper and things if it's placed elsewhere than in the facility itself, right? Cleaning and maintenance of portable toilets is important. And again, Amy may talk to us about that. And with that, I will just say thank you very much.
Nope. Did we just lose Callie?
We did.
Okay, I wasn't sure if that was me or all right. Well, she got to the end, so that was great. Thank you, Callie, and hopefully she'll jump back in. But with that, then we'll pass it over then to talk a little bit more about the surveillance programs going on here in New York State with Amy Robbins. So, Amy, take it away.
Hello, everyone. So we're going to just take a deeper dive into what goes on for those outbreak investigations and single case investigations. So I'm going to start by trying to share my screen. And what I'm going to do is I'm just going to go over some websites that offer some information, some of which have already been covered. So to start with, we have the CDC's page on cyclosporiasis. And I just wanted to start with, as we're talking about diagnosis, there's some great information here that people can look at on their own time. But to start with, you have someone who becomes ill and they typically have diarrhea for a couple of days. They finally seek healthcare and see their healthcare provider, and then they go on and get testing. and then it gets reported to the state. So in New York State, any laboratory test that comes back positive for cyclosporiasis is required to be reported to the local health department, and the local health department conducts an investigation. At the same time, any stool that's positive or slides that are available are supposed to be sent to our state public health lab for additional testing, confirmatory testing, and some genotyping. So that's kind of where I wanted to start. And then we also have sorry, I can't get to my other screen, so hold on one moment. So the big item that I wanted to bring up is that when we're doing our surveillance, let me try this again. So when we're doing our surveillance, the first step is that you call the healthcare provider, and then you call the individual case, because you want to figure out what their symptoms are as well as what made them ill. So for cyclosporiasis, there is a standard questionnaire called the Cyclosporiasis National Hypothesis Generating Questionnaire. And I have a little snapshot of it up here on the site that focuses just on the fresh basil. But what this questionnaire does is it asks for symptom information, it asks for travel information, which is very important for cyclo, and it asks for food. And this is all for the 14 days before the person became ill. So this is kind of one of our first stumbling blocks in the field of epi and surveillance is you've got somebody who might have been ill for a couple days, then they seek their healthcare provider attention, they submit a stool sample, the testing's done, they get the results, it gets reported, and we're finally calling somebody back and asking them, now that they're on day like 8 or 9 of their diarrheal illness, what they ate in the 2 weeks prior to their symptom onset. So it's going back for quite a period of time. And as was mentioned earlier, there's quite a few stealth ingredients. So our first step in New York State is you ask them about any travel exposure. Because if somebody was traveling internationally for those 14 days, we're not as interested in their food level of data, because we can only really do a lot of action on what's happening here in the US. So that's where we focus on domestic acquired cases. So if they've traveled internationally, we stop asking questions. If they didn't travel internationally, or they traveled internationally for part of their exposure period, we go through and ask all the questions that are on the cyclosporiasis hypothesis generating questionnaire. And so what you see here are the herb questions. So we might have a question about fresh basil. We might then ask what type of basil it is. And then if it was eaten at home or outside the home. And we want as much information people can remember as possible, such as brand names, such as packaging information, such as where they purchased it. Because one thing that we do have that's a unique surveillance tool for us to use is if a person can remember where they purchased it and they're part of a shopper card or a member plan at, say, Sam's or BJ's, or they have a card that they use to get the discounts, then we can work with that grocery store chain and we can find out what they purchased using that information from their card number. And then we can compare that to other cases as well. So that's a really novel and unique tool that we use on a regular basis for our cyclosporiasis investigations. But as you can see here, we're asking for detailed information about the herbs, and then it goes down and asks about fruit. And then it continues down to ask about some of the leafy greens, which is where we get into some of the information about how it's packaged. Now, one of the things mentioned earlier was the basil in the vinaigrette dressing. But one other thing is that a lot of people forget about what might be garnishes, whether they're on plates or whether they're in their drinks that they might have had in that 2-week time frame, which if all of you can think about what you ate 3 weeks ago today, it's a little bit hard to remember. So that's kind of our first step is that we look at these individuals. We try to get them to remember everything that they possibly can that they ate. And then we compare that to our other cases that are happening at the same time. So when we're doing that, we basically look at all of our cases and we say, okay, we're seeing an increase in, say, strawberries. And we want to know, is this what we would expect to see? So we have two ways of looking to see if we're seeing a true increase in a specific food item of interest. And one of those is using the Food Not Fast population survey. And this is a survey that was conducted from 10 sites across the country. And it interviewed individuals either by phone, or they did an electronic survey, and it was what they ate in the 7 days prior. And this happened between 2017 and 2019. And then the data is available online for the rest of us to go and look and see, like, these are what would be the background rate. And we can look at it by food groups. So here's the food groups, pick the fruits. And then you can also look at it by overall results or by certain characteristics, such as geographic location or season. So for cyclosporiasis, as was mentioned earlier and saw on the slides, there's definitely, we see more cases in the summertime. So here we can look at the food groups in the summer. So you can see here's the strawberries, and here's what the background percentage rate is of individuals we would expect to say that they had strawberries. And so this will help us figure out if it's a statistically significant food item that we're seeing more than we would expect to see. If we don't want to necessarily use the FoodNet Pop survey, or that question wasn't asked or available here, then we can look at our cases that we've had in the past couple of years, either look at cyclosporiasis cases or other enteric diseases that ask the same question. And then we can compare and see, well, are we seeing more in our current cyclosporiasis cases than we saw in the previous, say, 3 years or 5 years, or looking at all the other enterics in the same time frame period as well. So that's one way in which we can look at our data. We try to figure out what is statistically significant. For cyclo, the unique part of cyclo is that we rely on epi data more than we rely on the lab data. So for some of our other enteric diseases, we do a lot with what comes out of the whole genome sequencing. For cyclosporiasis, we rely on what comes out of the epi information. So to go on to the next, I just wanted to talk a little bit more about some outbreak investigations. So we saw earlier some summaries of the various outbreak investigations. And so what's available online is a website that has, on CDC's website, is information, more specific information about each of the outbreaks. So I brought up the fresh basil one, as this is one I worked on myself. That was back in 2019. And this one has some great information about the outbreak in particular. In New York State, we uncovered our cases through this by 2 events that happened locally. And individuals started to issue complaints back to the event, those who held the event, saying that people were ill, and did you know of others that were ill? And so then we were able to use those events, and it increased to more, and then to interview those individuals who went there to find out what specifically they ate that was served at the event. So that was slightly different than what we did for an individual case, because we were interviewing anybody who was ill at those events, not just those that were diagnosed with cyclosporiasis. And it brings us down to a smaller menu of food items on just what was served at the event. And that was another helpful way that we can do a surveillance. So we often think of those as subclusters, and those are going to give us more targeted information beyond our larger increase that we might be seeing across the county or a larger geographic area. So when these investigations are done for the outbreaks, and say we've identified something or we haven't identified something, they're all reported to the National Outbreak Reporting System. So I'm going to just bring up some of that information quick to share. And so for the National Outbreak Reporting System, there's a standardized form that is shared with everybody across the country, all the different jurisdictions that participate. And they share this information with CDC. And there's this lovely dashboard that's really got a lot of helpful information. And on here, you can explore the outbreaks that have been reported. So what I've done here is, using these filters, I've checked the boxes for every single type of outbreak that can be reported. I've selected cyclosporiasis from this long list of various organisms. And I've selected a year, and I went back to the previous 5 years, 2017 to 2021. And then what it shows here is a map. And you can go and you can over top of each state, and you can see how many states reported outbreaks. And then you have your statistics. There's 120 outbreaks between 2017 and 2021 for cyclosporiasis. If I back this up to 2009, there was 132. So we're definitely seeing more outbreaks in the last couple of years. Sorry about that. Then you have the number of illnesses and the number of hospitalizations and the number of deaths related to these outbreaks. Then this box here has overall statistics of every single outbreak reported to the NORS system. I know people were interested in water coming into this, and there is also ways that you can filter by water exposures and water types. However, I wanted to show for cyclosporiasis, you can see here's the outbreaks over the years. You can see the outbreaks per month, and the majority of the outbreaks are between May and through July. And the primary mode is foodborne. So we don't necessarily have a lot of waterborne outbreaks, but we definitely see the foodborne. And in this green color here is the indeterminate unknown. So those would be those individual outbreaks, say at an event where we weren't able to pinpoint exactly what food item was consumed. And here's a graph of the illnesses by year. So these outbreak investigations oftentimes are multi-state outbreak investigations, meaning that multiple state jurisdictions are involved, the CDC is involved, and FDA is involved. And you can spend a lot of time as an epidemiologist staring at the data, trying to find something in common. I know in New York State, for example, last spring, we had an outbreak related to a restaurant that was mostly people were eating there from a buffet. And it's just, there was just too many different food items, different garnishes, and it makes it very difficult, no matter how you look at the data, to figure out what people might have consumed and which single item is part of it, because we do have a lot of the stealth ingredients that we're trying to uncover. So I think that's basically what I wanted to cover for some of the surveillance. So I can turn it over to Amy next.
Great, thanks, Amy. Yeah, then we'll pass it over to the other Amy Kahler, who'll give us a little bit more of a perspective on the environmental side of Cyclospora and what CDC has been seeing.
Yeah, so I am going to switch gears and talk a little bit about wastewater testing for Cyclospora. It's something that we incorporated into one of our research projects that we did for the Center for Produce Safety. In 2020 and 2021, this was sort of a larger effort to assess whether Cyclospora was present in a particular growing region, and we're in Georgia, so in southeastern Georgia. We looked at environmental samples in the form mostly of irrigation water ponds, but then we were also interested in just understanding whether Cyclospora was even being shed by people in this growing region. And at this point, wastewater testing was not something that was, routinely done for pathogens. It kind of took off during COVID, and now there's a lot more emphasis on wastewater testing for things like coronavirus. But we, over the course of this 2-year project, we collected biosolids samples from the wastewater treatment facility in this area. We collected them from 2 different locations in the wastewater treatment process, simply because we didn't know the best place to go looking for Cyclospora, in a wastewater treatment facility. So over the course of those 2 years, we collected, I think, 76 samples. We tested them first by the 18S PCR that Callie mentioned earlier, and we got a bunch of presumptive positives. But because we work very closely with the parasitic disease group at CDC that developed these really great sequencing and genotyping tools for clinical samples, we wanted to go a step further and confirm all of our PCR positive results with these sequencing methods to see if we could, genotype any of the detections that we got. And what we found is that while based on the PCR results alone, we got about a 23% detection rate in all the samples we collected over these 2 years. But when you took that a step further and put those presumptive positive samples into like the sequencing and genotyping pipeline, it ended up that only 7 of those samples from those 2 years were confirmed as Cyclospora cayetanensis detections. And so is it possible for me to share a figure here?
Yeah, go ahead. Yeah,
Sure
you
thing.
can share your screen.
Do I do advanced sharing options? Oh man, this may be beyond my technical capabilities.
I don't know if you can do an individual figure. You might have to share like whatever program it's actually in.
Yeah. I'll just talk through it. But so we had, these 7 detections out of 76 total samples, and there were a few sort of notable things that we noticed or trends that we saw. So the first, obviously, was that, had we stopped at the PCR result alone, we would have said, 23% of these samples have Cyclospora, but because we took the step further to do the sequencing confirmation, we could say, oh no, it's 7 samples. So we would have grossly overestimated the occurrence of Cyclospora in this system over these 2 years, but also sort of winnowing out those maybe false detections allowed us to see trends in the data. And the most obvious one was that if you sort of plot the detections along the timeline, all of them occurred between July and September. So there's definite sort of seasonality to this. We can't really say what caused the seasonality, although, as Callie mentioned, most of the domestically acquired Cyclospora in the US is happening in the summer months. So that was an interesting thing that we could see when we sort of were able to confirm, which samples really were positive. The other thing was that we got more of those positive samples from the sample that was collected earlier in the treatment process from the aeration basin, where the sludge is settling out during the aerobic digestion. So that was something that can be taken into further, efforts. There might be places where, you're more likely to get the cyclo if you're interested in assessing, shedding in the community. So the upshot of all of this is, this was the first time we've done this. I think people are getting more excited about doing wastewater testing for all sorts of pathogens. And it's possible that it might be a tool that people want to use for Cyclospora. There are a lot of factors that go into play when testing wastewater that would need to be considered, but I think our findings can sort of are proof of concept, perhaps, that wastewater testing is something that may be a viable tool in the Cyclospora toolbox.
Cool. Thanks for that. Question on, do you think then, based on this one study, do you think then that the Cyclospora is underreported? I mean, the false positives might have been over-reporting in the system, but overall, does this mean we're not detecting it maybe as often as it's actually there? Or?
I think that's a definite possibility.
Mm-hmm.
Considering the long incubation period, a lot of people might not associate, what they ate 2 weeks ago with something that they're feeling 2 weeks later. And then it's hard to, if you do go to the doctor at all, to like correlate that back and sort of pin Cyclospora as the culprit of what you're feeling. There are also a lot of asymptomatic carriage of Cyclospora. I think you see that for sure in endemic regions. Where Cyclospora is endemic. How big of a issue is that in the US? We don't know. I think it's safe to say that something like Cyclospora is underreported just by the nature of it.
Oh, yeah, that makes sense. Well, thank you, Amy. And I'm gonna bring the panel back and open it up to questions from the group. So thank you, Amy, and Callie.